Hi, everyone, and welcome to episode 629 of the People Behind the Science podcast. Today, we are excited to be rebroadcasting our interview with Dr. Mariana Wolfner that was originally published on March 20th of 2015. Mariana is a distinguished biologist studying reproduction. Among her many scientific accomplishments, Mariana was one of the first to use recombinant DNA to isolate genes in the fruit fly Drosophila. She pioneered the use of complementary DNA hybridization to isolate the genes which respond to the steroidal prohormone ecdysone during metamorphosis. And she demonstrated key differences in the egg-to-embryo transition between Drosophila and mammals. In our interview, Mariana shares more about her career, her life, and her research. So, listeners, I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. Today, I am thrilled to be here with Dr. Mariana Wolfner. So thank you for joining us, Mariana. How are you today?
Fine, thank you. And it's really fun to be talking to you today.
Well, we are delighted to have you on the program. And listeners, I'm excited to introduce you to Mariana. So we'll start here. Mariana is the Goldwin Smith Professor of Molecular Biology and Genetics and the Stephen H. Weiss Presidential Fellow at Cornell University. She received her bachelor's degree from Cornell in Genetics and Development as well as Chemistry, and her PhD in Biochemistry from Stanford University. Mariana then completed a postdoctoral fellowship in the biology department at UC San Diego before she joined the faculty back at Cornell, coming full circle there. Now, Mariana has received many awards and honors during her career, including a Junior Faculty Research Award from the American Cancer Society, a Career Advancement Award from the National Science Foundation, a Lady Davis Fellowship, and election as a Fellow of the American Association for the Advancement of Science. Now, she has also received various awards from Cornell University for her excellence in teaching and advising. And Mariana is here with us today to share her own personal journey through life and science. So today, Mariana, we are excited to get to know you, both as a person and a scientist. So let's start off with just telling us what you like to do when you're not doing science.
Several different things. I enjoy spending time with my family. I have 2 kids now grown, but we still do things together when they're in town. We love to go hiking when it's not freezing here.
Right. Which it is right now, I'm sure.
Yes, right now it's really cold. And especially things like birdwatching and especially identifying the early spring wildflowers and that sort of thing. I love doing that kind of being out in nature and hiking, and that's been true ever since I was a kid. When it's cold, I like to be inside doing crossword puzzles and other puzzles. I just love doing that kind of thing. And baking things that are good and sweet to eat. Those would be my main enjoyments. Plus, I also am well known in my family for making terrible puns, and this goes on when I'm not in the lab, and a little bit here too.
Well, I am excited about this. Feel free to break out any terrible puns during our conversation today. I kind of like them. I'm not going to lie.
Do my best.
Well, Mariana, it's wonderful to get a sense of your personality, of what you like to do when you're not at work. You mentioned you like puzzles and stuff. Are you a Sudoku fan?
Yes, I am.
I am as well. They're kind of addicting. I'm not going to lie.
They are. And then just when you think you've figured out a good strategy, then suddenly that doesn't work and you have to figure another one out. Or, yeah, they're really fun. I'm glad to hear you like them too.
Absolutely. You feel like you're getting so close and everything falls apart. It's kind of a metaphor for science sometimes, right?
Exactly.
Well, Mariana, can you tell us next about your scientific side and how you describe what you do at work to someone who's not in science?
Sure. So my lab and I are particularly interested in reproduction and how it works at the level of molecules. So, reproduction obviously involves usually 2 individuals and a lot of social interactions between them. And we think that's fascinating too. And clearly it involves like an egg and a sperm. But in between those 2 levels, there are all these molecular interactions going on between the male and the female. And we're interested in the molecules that do this and how they work, and how male molecules work with female molecules, and how that whole thing evolves. And that's one of our major interests. And the other one also relates to reproduction, which is once an egg is fertilized, how does it switch from being a differentiated egg cell to being a brand new organism and brand new embryo that's going to divide and be able to make every kind of cell in the animal or plant. So those are the two general areas.
Well, you are answering some fascinating questions in the lab. And I know this second question still boggles my mind, how you go from this tiny little embryo to a whole person. And I know there's so many complicated molecular cellular things going on, genetic as well. So we'll be interested to talk more about your research projects as we go through the interview. But let's take a step back and talk about what motivates you. I know scientists have so much to get done in a day, so much on their plate. So can you tell us about a quote or a saying that you love to keep in your mind that keeps you motivated?
Yeah, I can't think of one that motivates me that often, but I can tell you one that I tell often to my students when an experiment doesn't work, that I guess sometimes has motivated me as well. So there was a professor here long ago at Cornell named Ephraim Racker, a biochemist, and I think the head of the biochemistry department here at one point, I'm not sure. But he was known for a whole bunch of sayings, and he had a saying that went something like this: it doesn't matter if you fall down, as long as you pick up something useful from the floor when you get up.
I like that.
So if I have a student whose experiment has just failed, I will sometimes use this saying, because sometimes the way it fails can tell you a little bit of something about how the process really works. Maybe your assumption was wrong, or maybe you were looking at it wrong. So that's a saying I like to think about.
Well, that is a fantastic motivational quote. And I think you can't help but fall in science now and then. And I think it is so important to get back up, but not only to get back up, but to bring some of those lessons with you. So fantastic quote to share with myself and our listeners today. Now, can you tell us a little bit about your journey? So you mentioned this one particular person you encountered who had lots of words of wisdom. Are there others who served as role models too, or people who you looked up to?
Yes, so I just barely knew Ephraim Racker. I knew his sayings, but not him so well. But one of the things about science for me is that, when I was growing up, you would see these movies about this solitary scientist who would go into the lab and, discover something important from a brilliant insight where they were all alone or with one assistant. And that's what I thought science was when I was a kid. And it turns out it's totally different. It's a much more social environment, and you're constantly shaped by the mentors that you have or by the colleagues that you meet or the collaborators. So for me, the inspiration has been several of my mentors. My very first research mentor was Jerry Fink, yeast molecular biologist who used to be at Cornell and is now at MIT in the Whitehead. And he found everything interesting. He made you feel like what you were doing was interesting and important. He could help you put things in context. And he was just really smart. And it just was something that watching him and hearing how he thought, seeing how he thought and how he moved from one topic to another was inspiring. Another one was actually Barbara McClintock. I got to know her when she visited Cornell. And then when I worked at Cold Spring Harbor as an undergrad for a couple summers, I got to know her quite well and went back to visit her frequently after that. And she was one of these people who was just determined to figure things out, even if they were complicated. And she had just a very interesting way of looking at science and answering questions and thinking about them. Those would be two. My graduate advisor, Dave Hogness, also was really good at just thinking broadly and running with things that were exciting, and I think had a big influence there as well.
Well, I love hearing about these specific characteristics or traits or qualities of your mentors that you've sort of picked up over the years or really admired in them when you were working with them. And I think that's so important as a scientist to have these people to look up to sort of learn the unwritten rules of doing science, being optimistic, being persistent, being curious and questioning things and putting everything in perspective. I think these are all really important things that they don't really teach you in graduate school. You have to learn it from someone who you're working with or who you admire. And I really like this point that you brought up of science being social and collaborative and collegial. And I think that's something people who might not be in science don't realize how much of a group effort scientific endeavors are.
Yes, it really is. And some of the people who have inspired me since my mentors will be collaborators or just colleagues I've met at meetings. Just talking to them and hearing their encouragement helps. And there have been times when those discussions have actually, in one case, more than one case, shifted what my lab did from one area to another, just because of inputs and collaborations with others. So you're right, it's really a social, or it can be a very social enterprise.
Absolutely. Well, Mariana, it's great to hear about these people who inspired you along your journey, but I'm curious about where this journey began. Can you take us all the way back to the beginning, to your earliest memories of becoming excited about science, and tell us what it was like and how you got started?
Okay, I think I was one of those sort of nerdy kids, I'm afraid, so I don't have one of these stories of brilliant insight suddenly hitting me.
I was a nerdy kid too. It's okay.
Oh, okay. Good.
I relish in it, yes.
Yeah, me too. There was a point where I didn't, but I've come to re-relish it again. So I think when you're a parent, being a nerd sometimes can be a little embarrassing with your kids around, but otherwise. So I grew up in New York City, and I spent a lot of time in the Natural History Museum there. And I still go back there whenever I go to New York. They had so many exhibits about life, and they had a big exhibit of a cell and its parts, and all these exhibits about how animals worked and what was out there in nature. That was one of the things that I remembered inspiring me even as a little kid. And going out into nature and on hikes and seeing the animals and plants and figuring out what they were was all things that really inspired me. And then I would do the nerdy things, like my mom would bring home, some chicken heart from the butcher, and then I would dissect it to see what was in it.
Wow, that's awesome.
So that was my start. And I'm not quite sure the link of that to what I do now, but I think I just became fascinated by genetics. It's such a logical and organized type of biology. You can predict the inheritance patterns that you'll get when you mate two animals or plants. And I just found that really beautiful and elegant. And that led me to major in genetics as a student. That led me into the lab and sort of on from there.
Well, I like how you're mentioning in terms of specifically genetics being a very logical and predictable sort of organized science to go into. I think a lot of people might not be aware that there's a lot more gray in science than initially you think when you're young going into it. You think everything's a fact, everything's established already. But I think when I hit graduate school, it was a shock at just how much we don't know.
Yeah, I agree with you in two different ways. One is there are occasionally people who say, well, it's all figured out now. We can stop. And every time someone says that, some incredibly brand new discovery comes out, be it epigenetics and how that works, or splicing or something like that, where it turns the whole thing upside down. And the other one is that when I have undergraduates in my lab, in some cases, it takes them a while to realize how much work went behind any sentence that they would read in a textbook. Mm-hmm. And like you're saying, how gray it is and how much has to be done to figure out what's actually real. And it's fascinating. There's always something to do, and it's not always as clear as it seems.
Right. It's exciting and daunting at the same time, I think.
Perfect. Yes, that's right.
Well, Mariana, can you tell us a little bit more about your own research and one of these projects that you are working on today that you are just the most excited about?
Sure, I'd be happy to. The pause was because we have two different projects that we're excited about, and I was trying to pick which one. Let me take one of the male-female interaction projects.
Sure.
So it's just fascinating to me that when animals mate and the male transfers sperm to the female, he transfers along with the sperm a lot of other molecules in the seminal fluid. And for a long time, those molecules were just thought to be things that nourish the sperm or help the sperm to make it to where they were going to go. And there are molecules there that do that. But there are also a lot of molecules that the male provides to the female that actually interact with the female's cells and molecules and affect her physiology, affect, for example, the muscular contractions in her reproductive tract, affect whether she can store the sperm he transfers to her. In some animals, like insects and camels, it affects whether she ovulates or not. And in some animals, like insects, they affect her behavior. And we are interested in what these molecules are and how they do this. And so we've been studying, for example, a molecule that triggers female fruit flies to ovulate the eggs that they contain after mating. And one of the things we're excited about, just a little while ago, a postdoc in my lab about a year ago, showed that this male molecule, which we named ovulin because it causes females to ovulate, mm-hmm. triggers a whole pathway that's already there in the female to make her muscles contract in her reproductive tract, pulling the egg out of the ovary, we think.
Interesting.
So, yeah, we're just fascinated. How is it that this male molecule knows how to work with the female molecules? And how did it evolve that a male could make a molecule that could turn on this pathway in females? And then a little further than that, a collaborator of mine, Yael Heifetz at the Hebrew University, and I have shown that the kinds of molecules that are affected by this particular male protein are spread throughout the reproductive tract of the female, and they change in amount and in position after mating, and that may help to coordinate all the different things that happen in the female, like the egg moving from her ovary, moving down the tract, moving to the place where it'll be fertilized, and then moving out. And that mating can be like a switch that triggers a whole process in this long organ of the female. So that's one of the things we're excited about.
Now, this is very interesting. So do you think these molecules are fairly well conserved across species, or are you finding different molecules in the different species you look at?
That is a perfect question, because when I started working on this, I expected the molecules to be conserved across species. And in fact, the female molecules that cause those muscle contractions are. But it turns out that the male molecules are the opposite of conserved. They're very variable between species. In fact, it's thought that they may have something to do with what keeps species apart reproductively, so that a molecule from one species of male fly will not probably, we don't know, but probably will not work as well in another species of male fly. So if the two species were to actually mate, which is difficult to begin with, the biochemistry then would not work and reproduction would not be stimulated. So that's actually a completely different, fascinating question of how does a male evolve to make molecules that manipulate the female, and then how do new molecules evolve in different species?
Certainly. Well, this is really interesting. And I think you're absolutely right here. You sort of keep getting more questions. The more discoveries make, the more questions you have, right?
That's right. Exactly.
Well, wonderful to hear more detail about one of these projects that you're working on that's really exciting in your laboratory. And I think we're painting science as a career to be a beautiful, fun, exciting adventure. And I know it is most days, but you do fall down sometimes in science, like we talked about with your motivational quote. You have to pick yourself, get back up. And I'm curious to hear about one of your own experiences, one of these challenging periods or struggles that you've had personally, and how you got through it.
Okay, I'll give you an example in a minute, but
Sure.
I will say that one of the things that keeps me motivated is that I work with students and postdocs, really wonderful students and postdocs, and in my mind, I'm doing this with them, and it helps to keep me motivated that we're working together. And so if something awful happens or something doesn't make sense, I always think about not only working with them, but I need to keep myself motivated and happy in order to keep them happy as
Right.
well.
You have to be the leader in the lab. Yeah.
Exactly. But it really does help to know they're there. So I'll give you an example. This one actually is an example of where a social thing came into play. It was in my first or second year as an assistant professor, and we had not started out studying the proteins I told you about a moment ago, but we were fascinated with the development of males and females. How is it that you start out with a fertilized egg and then it can either make a male or female, which are similar but actually very different? What are the genes that control the differences? And when I started my own lab, I was interested in what the genes were that were expressed differently in males and females, because they would help us figure out how they were controlled. And again, we work with fruit flies because they're pretty easy to work with and they have good genetics. And we started out looking at genes that were found at very high levels in one sex but not the other. And we were hoping to find genes that had something to do with behavior. So we found some genes, and I was going off to a meeting, the first meeting I ever went to as an assistant professor. And just before I left for the meeting, we figured out that the genes that we had found were expressed in a fruit fly tissue that's kind of the fly version of the prostate or the seminal vesicle, a tissue that makes seminal proteins. And my students and I were just distraught because, first of all, it's very far from the brain, so it didn't sound like it would affect behavior. And secondly, we figured that these genes were expressed in males simply because males had that tissue, and it wouldn't be that interesting to figure out how they were controlled because the control point would be the decision to make the tissue, not to make the proteins that we were studying. So this was like the day before the meeting, and
Oh no.
Yeah, and I gave my talk. And I explained how we were trying to study how male and female genes were controlled. And I ended up by saying, this method to find these genes works. But, sadly, the first genes we found were found in this tissue that's only in males. And a senior scientist, someone named Antonio Garcia Bellido, who was a very big person in the field, came up to me and said, "Do you know what this tissue does?" And I didn't, because I had just found this.
Right. You hadn't had time to look it up. Yeah.
Well, I hadn't looked it up. He said, "Well, you know what? This tissue makes these molecules from the male that go into the female and change her behavior."
Interesting.
And I was like, oh my goodness, this is incredibly cool. So the meeting was in Greece, and I called my lab from Greece. At that time, it was just by phone, expensive phone call. And I just said, "Go look up this tissue. And I think we're going to study this." And I got back, and everyone was just thrilled to work on this. And we shifted the lab over, and we've never looked back. These genes have been what I've spent most of my career on, and they are so fascinating. But I think if Antonio hadn't been there and hadn't said that, I'm not sure what we would have done. We would have probably kept trying to find other genes and probably found them to proceed with our original plan, but this was much more exciting.
Well, that is a fantastic story, Mariana. And I think a lot of scientists I've talked to have had these stories of sort of chance encounters, random conversations that you have with someone at a conference, with someone in your department that really changes the direction of their research. And in this case, sort of saved that project. I think that's wonderful to hear your own experience in this case and how your sort of stumbling, your failure turned into a success. So I love talking about success stories, Mariana. So I'm going to ask you next to share one of your recent successes. And this can be a big success or even just one of the smaller victories along the way.
Okay, this gives me a chance to talk about the other project, and I can do this fairly quickly. Our other project, our second project, where we're trying to figure out what it is that sort of turns an egg on to develop, to start as an embryo. So in this stage of development, it's called egg activation, when you shift from an egg to a young embryo, is not very well understood in any organism because it's very, very brief, and there's no new gene expression during this time, because what's happening during this time is that the egg's nucleus is finishing meiosis and the sperm's nucleus is coming in and then fusing with one of the haploid nuclei from the meiosis. So there's no new gene expression, which makes a lot of molecular studies more difficult. But there's one thing that was known in mammals like us and other vertebrates, which is that in those animals, when a sperm fertilizes the egg, it introduces some molecules in the egg which cause release of calcium from stored calcium in the cytoplasm. And this calcium release passes across the egg in a wave, and that sets off all the changes that start development. So we were curious to see whether something like this happened in fruit flies, because fruit flies allow us to study the molecules that would be turned on or off by calcium really well using genetics. But there's one little problem, or we call it a fly in the ointment.
Mm-hmm. I like it.
Oh, you wanted a pun, so there you go.
It's in there.
Thank you. One of our flies in the ointment is that the eggs of insects, including Drosophila, don't need to be fertilized to undergo this transition. And the best way to think about it is ants and bees, the males develop from unfertilized eggs. They're haploid. So clearly they didn't need a sperm to set them on that transition from egg to embryo. In Drosophila, haploid embryos don't live very long, but the transition also from egg to embryo, the start of it, also doesn't require a sperm. So we wondered whether calcium was still necessary. And we collaborated with the lab of Toshiro Eigaki in Japan. They and we discovered that we were asking the same question almost the same way at the same time. So we decided to work together. Some of his students came here. One of my students, Caroline Sartain, went to his lab. And we were able to show that, in fact, calcium does go up in fly eggs during this transition. And just like in all other animals where it's triggered by sperm, just like in those animals, it also passes across the egg in a wave, which is just beautiful to us to look at. We have a calcium sensor that fluoresces when calcium goes up, and we can see the fluorescence increasing and moving across the egg. And what's cool is, of course, no sperm is required in insects. So what's triggering the wave is as the egg moves through the female, it's getting squeezed in an environment that provides calcium from the outside. And the squeezing opens up channels that let calcium into the egg, and that's what raises the calcium level. So it's a completely different trigger, but the outcome is the same as in us and other vertebrates. So that's something we're very excited about. I watch these movies over and over again, another nerd-like thing, but I just think they're so pretty.
Well, it is very cool, and congratulations on this success. So when you got this finding, when all those sort of pieces came together, what was going through your mind?
Oh, wow. Best I could do. It just was so pretty, and it made so much sense. It was pretty hard to get there. I'd had a previous student, Vanessa Horner, and Toshiro had a previous student in his lab. We had tried many different ways to see it. We just had this feeling it had to be there, and we couldn't see it. And then finally we used a better sensor and we could see it, and it was like magic.
Very cool. I love that description. How, when you have that aha moment, that eureka moment in science, when you're finally getting all the pieces together, assembling all the data, and you see that big picture for the first time, I think it is like magic. And like you said, all the hard work that goes into it is finally being realized in that one moment. So exciting to hear about your success story there. And I think scientists keep so busy with the research that they do. And oftentimes it's hard to maintain good balance with all the other things that they're trying to get done in their life. And one of the things that I think often falls by the wayside is reading. I know a lot of scientists love to read, and you read a lot of literature, but you don't get to do a lot of fun reading necessarily. So I'm trying to get a book recommendation list assembled here. So can you add one to our list today, Mariana? Can you tell us about a book that you've read and really enjoyed that you think we should check out?
I can. Can I give you a couple of authors?
Absolutely.
I find that I like many different kinds of books, but I'm noticing that I tend to gravitate to books where the writing is beautiful and it also brings in some nature or biology. So something like Prodigal Summer by Barbara Kingsolver, or State of Wonder by Ann Patchett, or The Time Traveler's Wife by Audrey Niffenegger, which is a lovely book, and then suddenly starts talking about a fruit fly gene, which
really?
I was not. So those are three wonderful books that I've lost myself in and really recommend.
Well, fantastic recommendations. I will put them out there for our listeners if they are ready to lose themselves in the next book. And I'll have to check those out myself. The Time Traveler's Life, I hadn't heard of that one yet in terms of someone recommending it. So what was about that book in particular that you really enjoyed?
It was about a couple where the wife is just a normal person, but the husband travels back and forth in time and can basically jump back and forth at different times. And this turns out to be a hereditary thing, which is part of what was kind of fun. But it's just beautiful to see how he interacted with her as she aged, but he didn't necessarily, or he would come and see her when he was different ages. And it was just a really beautifully written and just a very interesting idea.
And of course, incorporating the genetics, very cool, the hereditariness.
Yeah, somewhere, just a little brief mention somewhere, but it was very funny to hear it.
Absolutely. Well, great recommendations, Mariana. And I know myself personally, I get most of my reading in when I'm on an airplane, when I'm going on trips. And I know scientists get so many great opportunities to travel. So can you think back on all the places that you've been for science, Mariana? And can you tell us about your favorite place? And what was it about this particular place that made it so special?
That's a really hard question in the sense that, as you say, scientists get to travel a lot. And that's been one of the really fun things about being a scientist, is getting to travel, to go to meetings in beautiful places, meet people who have become friends through science, talk about fun stuff, and be in these great places. So I have a little trouble, but I'll try a couple.
I'm going to make you pick one today, Mariana.
Just one? Can I do two?
You can do two.
All right. So I'll do two or three, but really quickly. The first is that there's a wonderful fly meeting in Crete every couple of years. It was actually at that fly meeting where Antonio Garcia Bellido made that comment that set our research on a different track. This occurs in a conference center that's associated with a monastery right on the coast, and it's a beautiful place, beautiful walks and swimming. You'll see people swimming and talking about their science, but it's just the most lovely place, and kind, happy people and wonderful food and everything. And so I've gone there occasionally for meetings, and they're always wonderful. I also spent a month in Israel teaching at the Hebrew University. And that was a beautiful place to be also, especially in the winter. But there was just so much history around us that in addition to the interesting science with colleagues there were just places to see that I'd always heard about. And it was just fun to go there and see them. And the final place was in Cambridge. I did a 2-week nano sabbatic with Michael Akam at the university there. And again, this was a place where you just felt the history. You could go to the place where Watson and Crick announced the structure of DNA. And it's just a lovely city. So that was my third favorite.
Well, it's wonderful to hear about these places that have really left a lasting impression on you. And interesting to hear that you're excited about the history of places. Are you a bit of a history buff yourself?
No, not really, I'm afraid. But I love to see it, see the places.
Absolutely. I'm one of those people, I don't read history books or necessarily watch the History Channel, but it's kind of fun to go to these historical places that have significance in science. I know a lot of people have gotten excited about the United Kingdom for, like you said, Watson and Crick, or just walking the halls of Cambridge where all those brilliant scientists have walked before them. So wonderful to hear about your experiences. So was there a particular site you saw in Israel that really meant something to you or was a memorable experience?
I think that the Dead Sea and the area around it was so different from anything I had imagined. It's very beautiful in a very stark way. And there is a very pretty gorge where there's a little bit of water called En-Gedi that has some very cool animals like ibex and hyrax, neither of which I'd ever seen in the wild. And those are particularly memorable. Plus, of course, a lot of the historical places like Masada and the Western Wall in Jerusalem.
Wonderful. I like to do a little bit of my traveling vicariously through the scientists I talk to. So great to hear about these places you've had an opportunity to go to. And I think in addition to traveling, one of the other great perks about being in the world of science is some of the great people that you get to work with, maybe collaborate with over the years. And I think all the labs that I've been in, at least, have their own little personalities and the traditions and quirks and funny memories that go along with them. So can you tell us today, Mariana, about one of these traditions you've experienced, or maybe just a funny science memory?
Sure. I'll tell you, being a Drosophilist, one of the traditions, not of my lab, but of my field, is that people come up with the weirdest names for their mutants.
This is true. I've seen many of them.
So genes that I don't work on, but genes like Methuselah, that was a gene that was involved in longevity, or Indy, which stands for I'm Not Dead Yet, which is another one of those, or Tin Man, which was a mutant that had no heart. And my lab, we've sort of missed our chance in most cases to make cute gene names. But we had one with collaborators in Switzerland, the Karch lab. They had found a strain of flies that we worked on with them where the males fail to keep their mates from remating, so that the female flies, instead of stopping to mate a lot after one mating, will just keep mating a lot. So that mutant was named Cocu, which is French for cuckold, because the males were constantly being cuckolded by their mates who mated with other males.
Gotcha.
It's just these kind of silly names, or the chance to have a little fun with your science, that I've always enjoyed.
Fantastic. And I think that's a really good point to bring up, just having fun with your science and these opportunities that you get, maybe a little bit tongue in cheek, just to have a little bit fun, put your personality into it and sort of leave your mark on the field in a humorous sort of way.
That's right. And they're easier to remember, the silly names.
This is true. You can always sort of tie the function with the name. I like it. Well, it's great to hear these different experiences scientists have had to bring fun into the lab. And I think it's really important to, for me at least, dispel some of these stereotypes out there about scientists and about how scientists are portrayed as very serious, sort of solitary people going into the lab doing their work. And I think, like we've talked about during our conversation today, it's not the case. Science is very social. It can be a lot of fun, and you can meet some fantastic people along the way. So in addition to talking about these experiences that you've had traveling, working with wonderful people, I love talking to researchers about the burning questions in their mind. So we've discussed some of the research that you're working on now, but can you tell me, Mariana, if we took away all the restrictions, if things like funding and staff and technology and feasibility were not an issue, what is the one question that you are just dying to know the answer to?
Wow, that's a hard one. I will play it a little close to home, but I will say that there are many fascinating questions out there that I could get excited about. But for me, I think I am so curious about how these male and female interactions evolved. How do pheromones evolve? How do molecules appear that one organism makes a message to another one? And how does that second one evolve to respond to it? How do pathways work that have molecules from two different individuals actually touching each other and work together? And related to that, how and why do they evolve in the ways that they do, in the ways that one animal will influence the behavior of another?
Well, all great questions. I think really interesting to think about your field in particular in the context of evolution and starting to stop and think about, how could this have evolved? So really, really interesting.
Yeah, we're just really fascinated by this. It sounds so simple when you think about it, but then when you stop and think, but how did it get there?
Right. Well, I love hearing about these big questions, and exciting to hear that you have the opportunity to work pretty closely to some of these questions that you're most excited about. And now we talked about some of the mentors who might have influenced you over the course of your career. And I know as a scientist, there are lots of opportunities to get advice, maybe a chance conversation at a conference or just from an advisor, a mentor, a peer, a colleague, a student. It doesn't matter necessarily where it's come from. You get some great advice. So can you share what you would consider the best pearl of wisdom, Mariana, that you've received in your career or your life?
I'm not sure from where I heard this, and I may have heard this from many people, but I think you just need to work on what you love and what excites you. And it may not excite, some of your colleagues, but if you think it's important and it excites you, you will do the very best possible job to figure it out, and it'll keep you motivated through the times that you mentioned before. That would be my main advice. I would say that it's also really important to find mentors and labs that will support you in this quest and that care about you and your development. And it's also okay to switch. Like if you thought this interested you and then you realize that something else interests you more, that's okay. Go for it.
Well, Mariana, I think this is fantastic advice, and maybe in particular relevant to some of our listeners out there who might be in graduate school, maybe 2 rotations in. They thought they were interested about this, but now they want to completely change and do something different. And I think it's okay, absolutely, like you're saying, to change directions. Or even if you're an assistant professor or later on in your career and you're going in a particular direction, but something else strikes you that's really interesting, it's never too late, I think, to change the direction your lab is going. So wonderful advice for our listeners out there. Now, Mariana, can you share one last piece of advice or maybe one last piece of inspiration with our listeners now coming from you?
This may sound fairly hokey, but biology is fascinating and wonderful, and there's always important and interesting questions out there. And I think people should try to solve them with enthusiasm and joy. There'll always be new stuff.
Absolutely. I like it, Mariana. Well said. Now, can you tell our listeners how they can learn more about what you're doing in the lab or more about you?
Sure. Probably the best way to find my lab is to Google my last name, Wolfner, Wolfner at Cornell. And that'll take you to our website, or feel free to email me. My email address is Mariana, Mariana .Wolfner@cornell.edu.
Wonderful. Well, listeners, definitely check out the projects that Mariana has up there on her website and send an email if you have any particular questions. And Mariana, thank you so much for joining us today and sharing a piece of your story on our show.
Thank you so much, Marie, for talking with me. This has been really a lot of fun. Thank you.
Well, I've enjoyed our conversations as well. And I want to take a moment to thank our listeners out there. Everyone, we will see you again next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.